Okay, so thank you for attending the webinar today. I'm Alex Mitchell. I'm based at the EBI here in Hinxton, and I'm going to talk to you about the EBI Metagenomics resource and how it can be used to analyze and explore metagenomic data. I've got a colleague here with me, Maxim, who will be happy to try and answer any questions as you go along. If you want to type them in the chat box, and he'll try and answer. Otherwise, there should be plenty of time at the end for questions and answers and things like that, so we can go into a bit more detail. So what I'm going to do is give you an overview of the EBI Metagenomics resource. I'm going to kind of try and explain why we exist, so some of the challenges we were set up to face. And then I'm going to talk about our data analysis pipeline and some of the algorithms, not in a great deal of depth, but give you an overview. And then talk about the website, the functionality, some of the things you can find there, and then some recent and forthcoming additions to the site. So some things we've added recently and some things which will be appearing in the next couple of months. Okay, so what I want to do is kick off with just a bit of a description of metagenomics. I'm sure most of you know what it is because you're attending the webinar, but essentially to stress that with metagenomics, you're covering a really wide range of environments. And what you're doing is going into those environments, you're sampling from them, and then you're kind of sequencing the collective genome, so all the genetic material you find there. But people are looking at really diverse things. So they're looking at really complicated environments, like rainforest soil, where you get all kinds of different bacteria. They're also looking at fairly simple environments, you know, human gut, where you don't get so many different bacteria. But it's a really broad kind of ranging field. There's lots of different things people are looking at. It's also quite a bit of an umbrella term. So people do lots of different experiments and they call them metagenomics. So some people take their samples and they size fractionate them. They pull out the viruses, or they pull out just the eukaryotes or prokaryotes. Some people don't care. They mix everything together and they just extract it. Some people are targeting particular genes, so such as 16S rRNA for to work out what bacteria and archaea are in their sample. That's not officially metagenomics, but it kind of gets lumped under the umbrella term. Other people are doing more traditional metagenomics, so whole genome shotgun sequencing, or they're pulling out the mRNAs, they're doing metatranscriptomics. So there's a really broad range of things people are doing with the data. But generally what they all do is they get the DNA, they sequence it, and then they perform analyses. And generally the analyses take two forms. So people are doing taxonomic analyses, so they're looking at things like 16S RNA genes for prokaryotes, 18S for eukaryotes. They're looking at ITS to try and work out what fungi might be in their sample, KEMA-based approaches such as Kraken for taxonomic analysis, and so on. And/or they're doing functional analysis, and usually this involves trying to identify what the protein coding section is doing, so trying to work out what's going on, and the kind of holy grail is to work out what bugs are there, what they're doing, and how they're doing it, to tie this all together. But actually, doing this properly is quite challenging. You have vast numbers of sequences, and I mean really, really vast. So some of the run files we're seeing now have got 500 million sequences in, and we can have tens or hundreds of these. Most of the sequences are missing from the reference databases, so you can't just look them up because they're not there. You have short sequences, which can be hard to characterize, so they might not have active sites which are kind of characteristic of the catalytic activity. They're getting longer, but they're still quite short. You can try and assemble these together, but you risk generating chimeras, where you're putting together sequences from different species. And people have kind of pointed out that metagenomics is a bit like a disaster in a jigsaw shop. You've got millions of different pieces from thousands of different puzzles. They're all mixed up together. Most of the pieces are actually missing because you're not sequencing them, and there's no box art to refer to. There are no reference genomes for these things. Also, you might find that some of the tools can have their own pitfalls and can lead you kind of into dark places. So there's no perfect tool for metagenomic analysis. The field is really broad, it's too big, there are too many people doing different things. So there are lots of different tools, hundreds of different tools. But you need to be aware that different analysis tools can give you different results. So if you use something like Chime, Mother, Kraken to look at taxonomy, you can get different results, different species popping out. Sometimes the same tools can give you different results depending on the version and the underlying algorithm. So for those people who may be terminally geeky, When PFAM upgraded from using the Hammer 2 algorithm to Hammer 3, there was a slight change in the way in which statistics were calculated, and this meant that some proteins that used to match some PFAM entries didn't match anymore. And we still have people mailing our help desk and trying to work out why this is, and one of the reasons was the underlying algorithm change. So that's really important to know about and to record if you want your results to be reproducible. Also, you might find that the same version of the same tools can give you different results depending on the reference database. So to give an example of this, I'm assuming everybody's kind of familiar with BLAST. This is NCBI BLAST, protein BLAST. We go there with our sequence we've pulled out of the plant. We choose the NR database at NCBI. We press search. We get back a set of hits. Our top hit is G protein coupled receptor from mouse-eared cress. We have an E value of 0, an identity of 100%. So we can be pretty sure that our query sequence has a very similar function to that, right? Well, if you go to the same data, to the same resource, so BLAST-P again, NCBI, but you choose a different database, so in this case, SwissProt, and you press go, so all the same settings, everything else the same, suddenly your top hit has a different name. It's a LAN-C-like protein with an E value of 0 and an identity of 100%. So are LAN-C proteins, LAN-C-like proteins, the same thing as GPCRs? Are they different? Which one of these is correct? Well, actually, there are no G protein coupled receptors in plants. People know about this. They publish papers on it. But the problem is there are issues with the NR database about who owns the data, who's responsible for making the annotations, who's responsible for cleaning up the misannotations. So it's really important to know not just what tools you're using, what versions of the algorithms you're using, and what reference database you're using, but also how are they maintained, how are they updated, and so on. Another challenge of metagenomic data is the sheer volume of data that you're getting at the time. So this slide is known as the most overused slide in bioinformatics. I don't really care. I think it makes the point really well. And what it shows is it's graphing up the price of sequencing versus Moore's law, which states that the amount of compute on a chip doubles roughly every 18 months. So you have this exponential growth in compute, and you can see the cost of sequencing was falling, and it was kind of tracking Moore's law up to about 2007, 2008, when next generation sequencing came online. And suddenly you've got this massive divergence. It gets really, really cheap to do sequencing, and it falls off and it keeps falling. So this is great. It means you can produce huge amounts of sequencing data, which is fine for doing things like metagenomics. You can go into really great depth. But it has costs. So this is a slide of a picture I've taken from a paper in 2011, and what they were trying to do was work out how much of a sequencing experiment the various elements cost and then extrapolate going forward. So they reckoned in about the year 2000, if you did a sequencing experiment, about 70% of your experiment, the cost was the sequencing itself. By about the time the paper was published, 2010-ish, they reckon that was split 50-50 between the kind of sequencing and the downstream analyses. And extrapolating further into the future, so 2020, they said the lion's share of the cost is going to be doing the analysis itself. It's not the sequencing, it's working out what all these sequences do. Okay. So there are other things to think about with metagenomic data too, especially if the data is going to be useful to the scientific community. And one of the things that it needs to be done is it needs to be archived. So it's no good having all this data which sits on your hard disk, which is usually precious, quite precious data. It might have taken a lot of time and effort to run, but you need to have it backed up. Other people need to be able to access it. And also, it's highly important to kind of annotate it with contextual metadata. So things like how was it sampled, how was it extracted, how was it stored, what the sequencing platform was, but also for metagenomics, things like where did it come from, what were the environmental conditions, the lat, long, depth, pH, salinity, temperature, clinical observations, blood pressure, status, illness of the patient, and so on. And the important thing about this is, if you adequately capture and describe your metadata, then you can ask really, really important and meaningful questions of the data. You can say things like, show me the microbial species which are found in a particular oceanographic sample. at particular depth, at particular time points, and then compare these to samples taken from a different position under the same conditions. So it allows you to really interrogate your data in a meaningful way. The problem is, we know scientists don't like capturing and recording this data. We know it's boring. What they want to do is science. But there are some considerations when you're kind of getting this data, and I hope you will get this data and record it, because it makes things much, much more useful. But the problem is, if you say to people, describe your sample, and just give them the text box, they can go a little bit crazy. So this is a slide taken from the European Nucleotide Archive, and essentially what they did was give people a text box and say, please describe the sex of your sample. Here there are about 88 different ways of saying that a sample is female. And you can see they range from kind of abbreviations to spelling mistakes to kind of really interesting descriptions. But essentially, this is what happens if you give scientists a kind of free text field and say, describe something. They'll never standardize. So this is one of the reasons kind of standardized vocabularies are important, and a way of making sure that people capture data in ways that are comparable. So those were some of the things which we had in mind when we came up with and we developed EBI Metagenomics. So I hope you've had a look at the site. It's a free resource for the analysis and archiving and browsing of metagenomic data. So we take the data, we archive it, we keep it safe, we back it up, and we make sure it's available. But we also do the analysis for free. There's no charge to use the site at all. Importantly, it's biome-agnostic. So some tools are kind of specialized for human gut, for example. We'll analyze data from all kind of size, all kind of environments, and we hope to provide powerful analysis, and as I mentioned, data archiving. So just to give you an overview of the process and what happens, essentially users submit data to us. The data goes into the European Nucleotide Archive. They offer tools to help transfer the data, so to move big files around, and to help capture and store metadata. So they have these kind of checklists with controlled vocabularies so you can describe things. We then take the data out of ENA and perform quality filtering, so we do QC. We then do sequence analysis using workflows that are versioned, so we know the reference databases, we know the algorithms. And then we put the data, the results on site so people can visualize, interpret, and discover them. So just to take you through some of the process if you wanted to submit data to us, the first thing you have to do is register for an account, and then you would upload your data and your metadata. This goes into European Nucleotide Archive. That can sometimes take a couple of days while files are checked to make sure they're corrupted and to get accession numbers and so on. But then we fish the data out, we analyze it through our pipeline, and then the projects, the metadata, and the results are all made available on our website, and they can either go into a public section where everybody can view them, or a private section for up to 2 years in the first instance to give you time to kind of write your papers, get your PhD, file your patents or whatever. That can be extended, but the kind of final idea is that everything will be made public available eventually. And this is quite an old slide, so it has some timescales on the left-hand side, which are a bit out of date now. But depending on the kind of amount of data we get, we try and turn things around within a week, a kind of week maximum-ish. But if we get huge amounts of data, obviously that takes longer. So that's how you get your data to the site. What happens once we get it? Well, as I mentioned, the raw reads go into the European Nucleotide Archive. We then take them out. We perform quality control. So we have some metrics to do with sequence length, sequence quality. Any that fail this are discarded. The rest, we take the process reads, we run rRNA selector to pull out the reads with rRNA. The 16S reads, so the prokaryotic section, we annotate with CHIME and green genes to produce taxonomic analyses. We don't give you eukaryotic taxonomy yet, but that's something that's coming soon. And then the sequences with rRNAs mashed out are passed to FragGeneScan, which produces the predicted coding sequences, so the proteins. We then try and annotate these with InterPro scan, which I'm going to talk more about, and then we provide a kind of functional assignment, or we also have sequences of unknown functions. So these are things that people are also interested in downloading and trying to work out what they are. Do they represent a novel biology? So I guess the most different part of our pipeline from other analysis systems is InterPro scan. So I'm just going to spend a couple of moments just describing what that is and why that's different to other approaches. So InterPro scan, what we're not doing is doing pairwise comparisons with things like BLAST, BLAT, Diamond, and so on. InterPro scan is different in that it uses protein signature databases. And so what I've done is kind of put a link at the bottom of the slide where you can go and find out more information about protein sequence databases that explains the background behind them, how they're constructed, and so on. But essentially what these are doing is taking a set of sequences that are related to each other, so they're a member of a particular functional family, or they share a domain, they're performing a sequence alignment, and then they model the pattern of conservation at specific positions in the alignment. And you can do this in different ways. You can use different approaches, so profiles, hidden Markov models, but essentially what you then do is you take your model, and if you have a new sequence, you compare it to your model, and if it matches, you say, yes, that sequence has that domain, or it's a member of that functional family. So this approach has been around for a long time. It's used by a whole variety of databases, including PFAM, TigerFAMs, Panther, ProSci, and so on. And the thing that the InterPro database does is it takes all these different member databases and puts them together. So it takes these member databases which are using different methods of producing their signatures. They're looking for different things. So some are interested in structural domains, others are looking at protein families, others are trying to predict active sites, binding sites, and so on, puts it all together. And again, I've given you a link at the bottom which has more information if you're interested in finding out. But essentially the idea is you take your sequence, you search against InterPro, you get back a set of hits which explain the kind of family to which your sequence belongs, with some functional contextual information. What can you infer for your sequence based on this family membership? Hopefully with some gene ontology terms, which are a kind of controlled way of describing the processes a protein is involved in, its function, and where it is in the cell. You get back hits telling you which domains your sequence possesses and what you can infer about the function of your protein based on those domains, and also active sites, binding sites, and so on. So that's essentially what we're using for our functional annotation. The reason we're using it is because we use it in-house to annotate the UniProt database. It gives us a lot of coverage, so it covers 80% of UniProt. It's a big source of gene ontology terms, and this allows us to have annotation consistency. Because we're using InterPro and Go for annotation, we can directly compare to proteins in UniProt annotated the same way. And also, entries are manually checked, errors are corrected. So I'm not going to say there are no errors in the database, but you should hopefully not get a situation like the one I pointed out earlier, where you have proteins erroneously annotated as having the wrong function. So that's kind of the background on our analysis pipeline and how we do the analysis. What I want to do now is quickly take you through the website and show you what you can do on it. I'm hoping you're going to come back to the site and have a play around afterwards. So this is going to be a kind of whistle-stop tour, and if you find things that you want more information on, then please email me. So this is a site. This is what it looks like kind of, I think this was grabbed a month or two ago. If you had private data, what you do is log in using this button here, but there's also vast amounts of public data that you can browse through and have a look at. So one of the things you can do is say, show me all the projects, or we have a search facility. So if you're interested in, for example, contaminated soil, you type contaminated here, you press search, and you get back a list of all the projects and the samples and the runs which are relevant to your search. And I'm going to explain the difference between those in a moment. So these are all contaminated somewhere in the metadata. We can filter down by biome just to get the soil ones. And here you can see we've got 39 projects which relate to contaminated soil. If we choose one of these, so this one here, we click through to the project. Here you can see that it's soil from a contaminated military base. We have a nice map showing where the samples are because the submitters very kindly gave us the latitude and the longitude in the correct format, so we can plot out where their sample comes from. And if we scroll down the page, we can see the different samples and the different runs associated with this project. We can see who submitted the project. We have links back to the ENA website so you can get the raw sequence files and so on. So We also have a version of the pipeline, so which version has been used to analyze the project, the runs, and so on. So if you click through here to the version information, you have kind of the full provenance and what's being run on your data. So which steps were run, which tools were run, which version of the database they're using. So the idea is that everything's traceable, you have full provenance, and so you can know if it's sensible to compare two different samples to each other and so on. So if we go back to our project page, if we choose a particular sample, like this one, we click through and we have some information, just high-level information about the sample, so the latitude, the longitude, and so on. But we can also have more detailed metadata. So here telling you what sequencing machine it was used, what depth it was dug up from, and so on. And essentially what you should do is consider sample to be the bucket. So it's the bucket of soil from which the various experiments are run. So you can have different runs associated with the sample. Sometimes people take the sample and they'll do 16S amplicon experiments and metagenomic experiments on the same sample. Sometimes people do technical replicates. So the sample is kind of the bucket of material. That's where the metadata, or most of the metadata, sits. Here we only have one run associated with this particular sample. So we click through, we can see that it's a metagenomic run. We have a series of tabs at the top which tells us more about the analysis results from this sample. So the first is quality control. So all samples should have this kind of quality control bar showing you how many reads we lose at various steps. So at our length filtering step, at our kind of trimming step, and so on. You can see how many reads make it through the process. For all runs analyzed using version 3 of the pipeline, you also get these kind of more detailed statistical analyses of the QC, so the read length, the GC distribution. You also get the nucleotide histogram position. And the idea of these is they're not just pretty pictures, they actually tell you meaningful things. So for amplicon data sets, you should see pictures like that one at the top. For WGS data sets, so whole genome shotgun, they should be roughly equal in their base calls. Actually, this one's got an untrimmed barcode at one end. Wouldn't matter so much for the kind of analysis we're doing on it, but if you wanted to assemble the data, that might kind of make things quite difficult. So these are kind of useful things to look at. So if we go back to the run, we also have a taxonomic analysis. So this is based on the 16S, which has been pulled out of the WGS dataset. We kind of default to the Corona view, which has kind of various steps down, but you can also look at different visualizations, so pie charts, bar charts, and so on. So that's a taxonomy. Functional analysis, we click through, you can see how many predicted protein coding sequences are in your run, how many of these have been annotated with InterPro. You have the complete set of InterPro matches, and you can click through to the InterPro database, which will tell you what it means if a protein has a particular domain and so on. We also summarize the top 10 hits in this kind of pie chart. And we also, as I mentioned, we annotate proteins with Go terms as well, and there's more information about Go at the bottom of the page here. But essentially, proteins have Go terms which can be very detailed. What we also have is what we call a Go slim, which is a way of kind of summarizing the specific Go terms into categories, and these are used for visualization. So you can see you get these kind of high-level visualizations of the functions and processes that the proteins in a particular run are involved in. The idea of these is that you can use them to quickly eyeball and compare different runs, and you can also do this using our online comparison tool. So we find the project in our tool, we choose some runs to compare, we can see the visualizations, and we can start to see if there are differences in high-level gene ontology terms between different runs, and that's a kind of place where we can then start to zoom in and get down to the protein level and try to work out what's causing these differences are, and if there are actual functional differences between runs. So you can do this on the website. We recognize that that doesn't address all users' needs, and people want to do lots of different things. So we also try and make all the files that we can available for download. So here you can get to the submitted reads for this particular run on the ENA website. You can download all the process reads with or without annotation. You can download the 16S to do your own taxonomic predictions, all the InterPro matches, the complete Go annotation, the slim Go annotation, and so on. And these are kind of quite small files. You can download them, you can take them away, and you can open them in other programs, editors, and so on. So that's essentially where we were at about 18 months ago. But what we realized was we were facing this massive growth in metagenomic data, and our pipeline really just wasn't fast enough to keep up. So we were processing billions of sequences per month, but we had an issue with the amount of data which was coming at us. So what we did was we got some help from some computer scientists, and we got these guys to come in and kind of look at how our pipeline was structured. And so you can see here our throughput actually went down as they started taking it offline and started making some fixes. And it took them a few months, but when they reconfigured the pipeline, put it back together again, suddenly we have this massive kind of uptick in the amount of sequences we can process. And what's really impressive about this to me is they didn't change any of the core algorithm for this. This was just plumbing, sorting out inputs, outputs, and so on. But it really boosted our ability to process data. And as a result of that, we can now put some really large-scale projects through the database. These are available online. So, for example, there's the Tara Oceans project. This was a really cool study where they sailed around the Earth. You can see their sampling sites here, and they took samples from all the way around the globe. We processed their prokaryotic data set. That's about 10 terabytes of sequencing data, so that's a huge amount of data. We're also getting their eukaryotic data set quite soon. There are things like American gut on the site. So this is over 80,000 16S amplicon data sets, 8,000, sorry, from various diverse individuals. And the idea is to try and tease out the connections between microbiome and health. We also have the UK Twins metagenome project, which is coming soon. That's about 3,000 samples. So we're able to cope with really large-scale data sets. You can see this is reflected in our growth. So last year, we kind of went a bit crazy. We processed vast amounts of data. Our current stats were at nearly 1,000 publicly available projects that you can go in and access. We've got about 77,000 runs, about 300 billion nucleotides, 150 billion predicted coding sequences, and about 50 billion InterPro scan matches. So that's a lot of annotations. It's also a lot of dark matter for people to download and kind of play about with as well. But this has really changed the way in which we have to think about the site and have to make it available to people. So to support people being able to come in and analyze the public data that's available there, well, one thing we've done is trying to break things up into biomes. So I'm sure you've seen these on the front page of the site. We tried to group projects into the biome to which they relate. So if you want to find all the soil projects, you can quite easily, all the marine projects, and so on. This is a hierarchical kind of listing system. It's based on the Genomes Online database. So if you want to find, for example, grassland soil or forest soil, there is that kind of level there. You can browse through and you can get it. The other thing we tried to do is make summary files for entire projects available. So every project now has this analysis summary tab, and this has a series of TSV files, so tab separated variable files, which have the matches to, for example, all the InterPro entries for all runs within a particular project, or the Go terms, the slim Go terms, the taxonomy, and so on. And these are kind of really manageable but quite nice summary files that you can, for example, plug into the Megan tool. You can even open it in Excel and graph things up if you want. So it's a way of kind of allowing users to get their hands on entire project summaries at once without having to process all the individual files. Another thing we've been working really hard on, sorry, I should just say, this allows you to do some really interesting analysis. So this is a nice analysis that people have done looking at different InterPro families, correlating them with different geophysical properties, so things like latitude, longitude, temperature, pH, and so on. And you can do this quite easily just by downloading those kind of data sets and then looking at the metadata. So one of the other things we've been working really hard on is trying to improve the search facility. So we've been indexing kind of huge amounts of data and kind of putting these together with EBI search. And the idea of this is that it covers questions that scientists really want to ask. So things like, give me all the antibiotic biosynthesis monoxide oxygenases in soil where we find actinobacteria because we know that they produce antibiotics which have been determined using metagenomics. That's actually possible using the resource. So you have to know at this point the InterPro entry for the particular family you're interested in. So we need to work on the functionality a little bit. But for example, if you were to search with this entry, which corresponds to that protein family, you can then narrow down your bacteria to say just, narrow down by organism, to say just projects with actinobacteria, to say just soil, to say just metatranscriptomics. And if you click through, you can see you have a set of search results with all the runs which kind of satisfy that criteria. So you can't yet get down to the sequences. That's something which we want to get to in the end, but that involves indexing kind of another huge tranche of data. But at the moment, you can get down to the particular run level, you can pull out the files, and you can sort of start parsing them and start pulling out the data. So you might have noticed I haven't really mentioned assembly very much. When we started the site 4 or 5 years ago, there was kind of really limited choice in the kind of assemblers that you could use for metagenomic data. There weren't many of them, and they were very computationally expensive. So it cost a lot of compute to try and assemble data. And actually, we found we did quite well just annotating the raw reads. So this is a graph which shows a reanalysis of some data produced by Hess in 2011, and he'd taken some metagenomic sample from cow rumen. He'd been looking for things which were involved in carbohydrate metabolism. He'd annotated them using PFAM, and he'd done assembly. We performed a similar analysis using just the raw reads, and you can see we get fairly decent tracking between the two. So his results are in green, ours are in red. We think we're doing okay. However, things change and things move on. So one of the things we're looking at now, as more assemblers and better assemblers come online, is maybe we can do some assembly of some of the data that we have. So this is some kind of pilot work we're looking at the moment, looking at different biomes because there are differences in kind of computational needs according to how complex a biome is, and looking at things like input sizes and so on. What's interesting here is we know human gut isn't very complicated, so actually it seems to plateau off. We don't need so much more memory, we don't need so much more compute time as the input file goes up, because it's fairly kind of not particularly diverse sample. With things like soil, we seem to have this linear relationship whereby they just get bigger and bigger and bigger, and we just need more and more memory and more time to assemble the data. So that's something we're looking at. We're trying to work out the best way to support assembly, and I would imagine that limited assembly will appear at the site in some time in the future. We're also looking at other taxonomic analyses. So I mentioned we're going to start offering eukaryotic analyses fairly soon. We're also looking at different systems to perform taxonomic analyses, so things like p-placer, KMA-based approaches, things like Kraken, Keiju. So I'm spending a lot of my time looking at plots like these, which show how you get different mappings to kind of family, genus, and species level if you vary things like e-values, confidence scores, and so on. And we also have SIMCA coming up. So SIMCA is a really nice tool which performs kind of analysis of KMA spectra, and what you're able to do with this is place related samples, related runs to each other, and split out ones which don't appear to be related. So it's another way of kind of performing comparisons, and we hope that that's going to appear on the site in the kind of next few months or so. We'll probably have to have a follow-up webinar where we explain the kind of new functionality. So that's pretty much from me. I just want to sort of point out that we're a fairly very small group who are working on this. So Rob Finn is our kind of group leader. There are two guys who work full-time, Hubert and Maxim, who are developer and bioinformatician, and the other guys who are involved in kind of web design and supporting the pipeline and so on. And we also work closely with our friends at ENA who support all the submission side of things. I should also acknowledge our funders. Okay. Well, thank you for coming. Thank you for listening. I hope you use the resource and find it useful. If you want to find out any more, then please feel free to email. I think my email is on the front page of the slides, so please just send me a line.